Okay, good morning, everybody. Welcome to the lecture on histology, and feel free to use these yes, no buttons you might seen in the participants window if I will ask some question. And you can also use the go slower or go faster, because this my intention is to briefly go through the key points of the lectures you missed during the past weeks, and then we will talk about the new topic, which will be the formation of blood or hematopoiesis. So my first question will be, can everybody hear me? Is it okay?
Yes.
Okay, and you can use these buttons, yes or no. I see immediately how many students are answering. Okay, so I need to know what do you know or what you don't know or don't understand from these topics from the past 3 or 4 weeks. So let's make a brief survey of this, and please use this code to go online, and there'll be a few questions for you. I will perhaps add the code also to the first questions I might have, if somebody did not manage to make it in time. But it's the same address we have already used. So this should be brief. Please answer what do you believe is the correct the statement here. Either you can scan this code or go to this web address, paulev.com/histologiapl007. You don't need to register there. You can skip the registration step and answer anonymous as anonymous guests. And if anybody got technical problems, just feel free to use the chat window, okay? Okay, I already see some results, so there are it works technically. So I'm waiting for more students to provide answers. If you go to this address, to this web page, you will have the opportunity to submit your opinion. And my idea is to go through like 11 or 12 of these questions. Then I have the idea what do you know and what you don't know, and I will adapt the rest of this lecture accordingly, because we are starting after a 3 weeks break. And as you know, the medical education does not forgive any gaps, so we need to be perfectly sure you understand the topics we have missed. So I need this as my feedback, what you know and what you don't know. So I was hoping for more answers. I see only two students submitted. Oh, you are not able to enter.
Yeah, we don't know why, but somehow it's not running.
Okay, what about now? Perhaps I forgot to activate the, yeah, that's it. Is it okay now?
Yeah.
Okay, thanks. Thank you very much. Sorry about that. Okay, that's decent. 25 answers, 27, 30. So I will lock the answers now. The DNA is not, the mitochondrial DNA is not wrapped around histones. The mitochondrial genome has no histones. The DNA is maternally inherited, that's true. Mitochondrial DNA does not have repair mechanisms comparable to nuclear DNA. It's very simple DNA. And it's also true that most of the mitochondrial protein, including the respiratory chain proteins, are coded in the cell nucleus. That's why we call that organelle semi-autonomic. It's dependent. It depends on many genes coded by the cell nucleus. Another question asking about the basophilic structures. It's a very frequently used term in histology. Basophilic means dark blue or dark purple. Now, which of these structures would appear dark purple or dark blue? That means basophilic. And why would be that? It's very practical to know that, because you can read the basic chemistry from the staining of your routine sections. Okay, we got like we got 30 answers. I'm looking at this now. Now, which structures are basophilic? Cytoplasm of muscle cells and muscle fibers, it's eosinophilic. It's reddish, actually. Yeah. So A was not correct, but the cell nuclei and nucleoli are typically basophilic because they are made of nucleic acid, which binds these basic histological dyes. From the same reason, also, this is correct. The rough endoplasmic reticulum is also basophilic. Why do we care? Because the cells that are specialized for producing proteins, they have very rich rough endoplasmic reticulum, because the translation occurs there, and they appear, the cytoplasm often appear as basophilic. That's why we care. Red blood cells, as we'll see today, are not basophilic. In the opposite, they are eosinophilic. They are pink to orange or reddish. And the cytoplasm of hepatocytes is also not basophilic. It contains numerous mitochondria, but they make it eosinophilic. Okay. Another problem. We go to the epithelia. Simple columnar epithelium. Very simple question. Could be fast for you and easy. Where do we find simple columnar epithelium in the body? If you, why do we ask that? Think about a slide you will get during the credit exam, and you will identify the simple columnar epithelium. Yeah, anybody should be able to do that. Now, which slides do you start to think about in your diagnosis? You need to know where this is in the human body and use this information for diagnosing the slide. Okay, I will lock the responses soon, like now. Let's have a look. So, sorry. Simple columnar. The small intestine and large intestine would be fine, but the esophagus is the problem. Esophagus is aligned with a different epithelium, namely the stratified squamous non-keratinized epithelium, which continues from the pharynx, right? So A was not correct due to the esophagus. Stomach and gallbladder, the majority got the right question. Both have simple columnar epithelium. Not the oral cavity, that's lined with stratified squamous non-keratinized epithelium, mostly. And nasal cavity has pseudo-stratified columnar ciliated epithelium. So this was not correct. The oviduct has simple columnar, and the uterus also has simple columnar epithelium. So this was correct. And distal tubules of kidney, nephrons, they have cuboidal epithelium, simple cuboidal. And alveoli of lungs definitely don't have simple columnar. Columnar is tall. Yeah, you are breathing through the alveoli of lungs. That's where you have the most flat cells in your body. It's simple squamous epithelium mostly. Yeah, the type 1 pneumocytes. Now the embryonic origin, which also belongs to the thinking about epithelia. Where do they come from? We are asking about 3 germ layers, ectoderm, endoderm, and mesoderm, and your task is to find the correct combination of structures and organs that develop from these embryonic germ layers. We know that epithelia of the human body are actually derivatives of these embryonic epithelial germ layers. So let's discuss this briefly. The embryonic origin belongs to one of the characteristics of tissues in general histology. Okay. So let's take a few more seconds. Okay, so ectoderm. Ectoderm differentiates into epidermis. Yes, I'll log it now. That's correct. Nerve tissue, yes. It comes from a specialized area of ectoderm called neuroectoderm. But kidney is the problem here. Kidney differentiates from mesoderm, not ectoderm. And thyroid gland, the site of embryonic origin of thyroid gland is the thyroglossal duct, the remnants of which you know as the foramen cecum on the linea terminalis between the body and root of the tongue. And that is already the endodermal part of the pharynx, where the thyroid gland develops from before it descends to its final position on the thyroid cartilage. So thyroid gland is endoderm, not ectoderm. Second option, endoderm gives rise to esophagus. Yes, everything starting with pharynx, esophagus, stomach, small intestine, large intestine, to the ending with the rectum is from endoderm. This includes, of course, the glands that are actually evaginating from the intestines, which is liver and pancreas. Yeah? So that's also correct. Intestine, yes, and the urinary bladder, yes, because urinary bladder develops from an organ called cloaca, which is at first a common opening of the GIT and genital and urinary system, and cloaca will become separated to the dorsal rectum and the ventral urogenital sinus, from which the urinary bladder develops. So this was correct, this one. Mesoderm. It differentiates into the lining of peritoneal cavity, yes. If you know the term mesothelium, which is the simple squamous epithelium lining the peritoneal cavity, the meso in the word mesothelium reminds you of the fact that it really comes from the mesoderm. So are the gonads. There are also the epithelial component of gonads is also mesodermal. but not the lungs. The lungs are on the end of the tracheobronchial tree. And if you ever wondered why trachea, that is attached to larynx, appears to be a branch of the pharynx, that's because it originated embryonically from the embryonic esophagus. It's an outpocketing of the GIT actually. So therefore, it also shares the same embryonic origin, which is the endoderm, as we already know. Okay, so this was correct. This one. Let's move forwards. Asking about mucus cells. So this is testing whether you know what mucus cells are and where they could be found in the human body. Perhaps you have, or definitely you have also seen some slides in your practical classes, some slides with the mucus cells. So you already have your own experience with that. And if you know how the glands are distributed, it's really helpful in the diagnosis of histological slides. That's great. Let's have a look. Okay, I'm locking the responses right now. Root of the tongue. This is correct. Actually, if you look at the root of the tongue, you can see the stratified squamous non-keratinized epithelium on the surface. The lamina propria that is heavily infiltrated by lymphocytes and lymphoid follicles. That's why the root of tongue is also called sometimes the lingual tonsil. The tonsil being an area of mucosa infiltrated by immune cells and serving the immune function. And if you go deeper, you got the muscle of the root of the tongue, and among the skeletal muscle fibers, there are mucous glands distributed, called the mucous glands of Weber. Spelling is W-E-B-E-R. So this was correct, right? Well, respiratory bronchioles don't contain glands like bronchi had, and definitely not alveoli of the lung. You want to breathe through the wall of the alveolar septa, and it would make actually no sense to produce some mucus in there. The only secretion we got there is the so-called surfactant, which reduces the amount of mechanical work necessary for expansion of the lung alveoli. But this is not a mucus. It's a mixture of lipids and lipoproteins. So this was not correct. Okay. Urinary bladder, no, we don't have any mucous glands in the bladder. There are no actually mucous glands or no glands at all in the vagina itself. So I need to disappoint you. This was not correct. There is a secretion that comes out from vagina, but the source are mucous glands of the uterine cervix. Yeah, the cervix of the uterus, it does have mucous glands, and the secretion comes to the vagina, but it's not produced there. If you look on the histological slide of vagina, you will find no glands there at all. And now, last option. Mucous cells would be inside submandibular and sublingual glands, because these both are combined seromucous glands. They have both mucous cells and serous cells. Okay, but the parotid is the problem here. The parotid gland is entirely and purely serous gland. It has no mucous glands, so that's why the last option was not correct. So actually only the first one was correct. Okay, next one. Now the questions will go more complex. We're moving from the simple to like more advanced questions. which cells do you expect the granular endoplasmic reticulum to be well developed? So you need to understand what's the function of the granular endoplasmic reticulum, and you need to understand what is the function of these cells we are asking about. And you should be able to put these two things together. That's the key how to answer this problem. And why do we ask actually questions like that is because it kind of mimics the way of thinking if you're looking on the slides under the microscope. Let us say you already know that the cells with well-developed granular endoplasmic reticulum have basophilic cytoplasm due to that organelle. So if you find a basophilic cell, it mostly tells you what a cell is doing. And that's our aim in histology, yeah, to combine the shape and shapes and colors you see with the microscope with understanding of the function of the tissues and organs. So you're actually looking for cells that have that are specialized for production of proteins. So let me check your answers. A few seconds before we move forward. Okay. Let me lock this. So we are looking for cells specialized for producing proteins because we all agree granular endoplasmic reticulum has ribosomes where the translation of proteins happens or occurs, right? So goblet cells are definitely not the answer because goblet cells are actually mucus producing cells. You can find these, yes, on the intestinal villi and inside the crypts, that's correct. But goblet cells are not specialized for producing proteins. They produce mucus. And mucus chemically is made of complex polysaccharides, not proteins. So this is not correct. B, that's correct because exocrine pancreatic acini are aligned with typical serous cells with basophilic cytoplasm producing proteins of the pancreatic juice, such as proteases and lipases and other digestive enzymes. So that's definitely correct, and that's helpful. Lytic cells among the seminiferous tubules, yes, they are there. Lytic cells are the producers of androgens, of male sex hormones. And you should know that these are steroid molecules, yeah, not proteins. And the synthesis of steroid molecules occurs mostly on a smooth endoplasmic reticulum, not on the granular. So Leydig cells are not the correct answer. Moreover, the Leydig cells accumulate precursors of the synthesis of steroids, which are lipid molecules, mostly cholesterol droplets in the cytoplasm. So they have actually eosinophilic to light cytoplasm because the chemistry of the production of steroids is different than the chemistry in organelles serving for protein production, which the question was about. So this is not correct. Epithelial cells of the adrenal cortex, that's similar to the Leydig cells. Adrenal cortex is producing steroid hormones, mineralocorticoids, glucocorticoids, androgens. And again, these are not proteins. Brunner's glands in the submucosal duodenum, yeah, they exist, but you should know that these are actually mucus glands. They partially help to neutralize the acidic chymus coming from the stomach, which with a pH like 1.5 or 2 needs to be rapidly neutralized to pH 7 in the duodenum, which is then the optimum for the digestive enzymes coming from pancreas. And there are basically two ways how to quickly neutralize the acidic chymus coming from the stomach. First is the pancreatic juice with its bicarbonate buffer solutions, and also these other Brunner's glands that protect the mucosa of the duodenum from being digested by the aggressive enzymes and being damaged by the hydrochloric acid coming from the stomach. So Brunner's glands are mucus. That's why I don't have endoplasmic reticulum well developed. Okay, we're moving to the connective tissues and asking about the macrophages. Which of these would be, according to your opinion, the main function of macrophages? Which of these processes are typical for macrophages? Okay. So let's have a look on your responses. I'll be locking the poll soon. So some students believe that is the production of immunoglobulins, which are antibodies. This is a specialty of other connective tissue cells called plasma cells. We'll revise that briefly after this poll. Endocytosis or phagocytosis of lysosomes. Well, I understand that hearing the word endocytosis or phagocytosis in the context of macrophages sounds like a catchy answer. But if you think about the meaning, it doesn't make sense. Lysosomes are intracellular organelles. They are membrane-bound vesicles that are formed inside the macrophages from the Golgi apparatus, and that's where some aggressive enzymes are stored, and they are prepared to digest particles or microorganisms that come inside that are phagocytized. So actually, no macrophage performs phagocytosis of lysosomes. Yeah. So don't think about the meaning of the answer. Release of histamine and heparin as part of the inflammatory response. This is more task of cells that contain connective tissue cells that contain histamine and heparin granules, and these are called heparinocytes or mast cells. So not macrophages. A production of highly reactive oxygen species and proteolytic enzymes, that was the correct answer here. The highly reactive oxygen species are very aggressive forms of oxygen, such as superoxide ions or peroxide ions. The macrophages are producing special enzymes to generate these highly reactive molecules. They have a peroxidase and they have superoxide dismutase and other enzymes, and also proteolytic enzymes. So this is how the macrophages are attacking the microorganisms and other particles they have eaten. So this is correct. It's even called respiratory burst, the activation of macrophages to do this. It's called respiratory burst. Exocytosis of procollagen and proelastine into the extrasolar matrix, that's a task for fibroblasts. So this was correct here. Next question deals with the extrasolar matrix, and they are 5 statements. So let's read the statements and discuss which would be correct. So again, this is already a more complex or advanced poll. It's testing 2 things. First, if you know the right words and their meaning. And second, if you know the structure or the form of these components of the extrasolar matrix. Okay, that's fine. I see over 35 answers in the poll. That's nice, even 40. Okay, so I'm locking the poll now. Elastic fibers are made of elastin core, and it's surrounded by microfibrils of fibrillin. That's perfectly correct. That's true. Collagen fibers are grouped together in a form of large double helices. That is incorrect, for 2 reasons at least. First, the typical arrangement of the basic structure of collagen, which is called tropocollagen molecules, is a triple helix. And second, collagen fibers, which are already like 1 to 2 micrometers thick structures you can see in the microscope, they are not grouped together to form some helices. And the opposite, they are made from many tropocollagen molecules, each of which is made of a triple helix of individual collagen molecules. So the hierarchy of these terms was the opposite than suggested in this option. Glycosaminoglycans, such as hyaluronic acid, so far so good. Hyaluronic acid is one of the glycosaminoglycans, which are usually highly hydrophobic. Well, hydrophobic means the molecules repulse water molecules, okay? However, hyaluronic acid is doing exactly the opposite. It binds lots of water, amounts of water. Each gram of hyaluronic acid is able to bind hundreds or grams of water. The molecule of the hyaluronic acid contains so many hydrophilic chemical groups that have their water solvatation envelopes, and the opposite. Hyaluronic is one of the most hydrophilic substances we got in our connective tissues. If you recall, for the same reason, it's used in the cosmetics to increase the hydration of the tissues to bind water in the tissues. It also, why a doctor should care about this, it's not only because of the cosmetics, but it's about understanding the flow and diffusion through the tissue fluid. The cells in our connective tissues, they are actually bathing in a fluid called tissue fluid. That's where all the nutrients from your blood plasma are released to. That's the fluid that immediately surrounds the cells and where all the metabolites are produced before they are drained by lymph or venous blood. And the diffusion in this tissue fluid is strongly dependent on the water. That's why dehydrated tissues and organs fail to work. Oh, it's one of the reasons. And hyaluronic acid is one of the components that keeps water in your tissues. Unfortunately, as we're getting older and older, we produce less hyaluronic acid and our tissues get less hydrated. So remember, forever, hyaluronic acid is highly hydrophilic. It binds water. Glycoproteins, the molecules of which contains mostly polysaccharides surrounding a small protein core. If you hear a word glycoprotein, even the composition of the word suggests that it's a protein, which is the main component here, that is modified by some saccharide component by a process called glycosylation. So there are shorter or longer oligosaccharides or saccharide molecules that are attached on the main protein core. So it's the opposite here. Yeah, the protein component is the main one. This would be a definition of proteoglycans, by the way. Yeah, this one. That is made of mostly polysaccharides. Multi-adhesive structural glycoproteins, they exist. Fibronectin and laminin are good examples of these, which interconnect the cells with the extrasolar matrix. Perfect. That's like kind of a definition. I'm glad you knew it. That's correct. Now, I will prepare. Oh, sorry, give me a second. I forgot to include a slide here. So I need to fix this. I need to fix this in a second. There was supposed to be a slide there, and it's not. And it will be this one. It will be this one. So now you should see a tissue or some cells, and type in anything that comes to your mind. Where this might be come from, what are the cells, what structures are you looking at? If you see in the microscope anything like that, what could that be, possibly? Okay, I see already some answers. Adipose tissue, which is correct. Liver is not correct. We see actually two types of adipocytes here. These are unilocular adipocytes in the white adipose tissue, and these are multilocular adipose tissue cells, where the lipids are distributed in a large amount of lipid vacuoles. That's the brown adipose tissue. So we do see both types of adipose tissue side by side. Okay, very nice. Most were correct. Now we approach the blood topic, and this requires some understanding and thinking, which exactly what we want you to do. Which statement about the blood count and differential count of leukocytes would be true? So think about these statements. So this question is testing whether you know the words that are used here: eosinophils, Arnett's count, reticulocyte count, platelet count, and neutrophilia, and so on. And secondly, I'm testing whether you understand what this is good for in medicine. Of course, in reality, it would be much more complicated. So this is a simplification for first-year students, but I'm sure you will deepen your knowledge in that field in other subjects in the future. But this is actually the context why we are discussing these terms in the histology of blood. So I'm curious about how you understand these words. Most of it was contained in the previous lecture on hematology that is already hanging on the Moodle platform for a week or so. Okay, so I will be happy to look on your responses. How do you responded? And I will log the poll. So you would expect lower count of eosinophils in patients with asthma, type 1 allergy, or in patients with helminthosis, which is a parasitic disease called by helminths. You should have linked eosinophils with asthma and type 1 allergy, but in the context of a higher count of eosinophils, because asthma is like kind of eosinophilic inflammation of the respiratory passages of the bronchial tree. The type 1 allergy, anybody knows the symptoms, is also linked with increased activity of eosinophils. And the parasitic diseases, the parasites in your body are actually attacked and surrounded by eosinophils. So it's the opposite. You would expect higher count of eosinophils under these circumstances. Second, vitamin B12 deficiency may result in left sheet in the Arnett's count. So what does it mean? The vitamin B12, also known as the cobalamin or cyanocobalamin, is one of the vitamins that is necessary for proper erythropoiesis, for maturation of red blood cells. So without B12 vitamin, the patient suffer from a defect in the formation of red blood cells, erythrocytes. The erythrocytes are large, but they don't work well. But here, the left sheet in the Arnett's count, that's something different. Arnett's count is a sequence of neutrophilic granulocytes maturating from the younger ones to the older ones. So these two things, as far as I know, they have nothing in common. So this is not correct. C, increased reticulocyte count can be found after acute blood loss. So first you need to understand what reticulocytes are. In this context, not to be mistaken with the reticular cells of connective tissue. When it comes to blood, the reticulocytes are the first young forms of red blood cells that might appear in the peripheral blood. There should be like only 1 or 2% among the circulating red blood cells, but they occur there. And this is correct, because after losing red blood cells during blood loss, your body is trying to replace this loss by releasing more of these young forms into the peripheral blood, and it accelerates the hematopoiesis. So this is exactly why you need to know about reticulocytes. It indicates there might have been some acute blood loss recently. A patient has 30,000 platelets per cubic millimeter. You can perform surgery. You don't care about some adding more platelets. Okay. Of course, this the normal platelet count is like 150,000 to 400,000 platelets per cubic millimeter. Patients who have such a low platelet counts would have unstoppable bleeding if you would start surgery. So they would literally bleed out on the operating theater. You should not perform surgery without adding more platelets. Neutrophilia is a typical sign of acute bacterial infection. That's perfectly correct. Neutrophils are first on the side. They are highly mobile, active granulocytes. They are also macrophages. They have the ability to to perform phagocytosis. And that's the first line of defense when it comes to bacteria. I have another picture here, and it will be a different slide, namely this one. Name what you see, please, and I will clear the previous responses. So if you see something like this under the microscope, what it might be. Oh, I'll show the responses. What about how would you call the tissue? In which organ the tissue might be, right? So erythrocytes, cartilage, chondrocytes. Okay, this is cartilage. Does anybody know what type of cartilage is that? The cells are chondrocytes. Look how the cells are grouped into groups that are called isogenous groups. And I'm testing now if you know what type of cartilage would it be. Look how the extracellular matrix, how it has relatively homogeneous appearance. You don't see the fibers there. Well, they are there, namely collagen 2 type 2 fibers, but they have the same refraction index like the matrix, so you don't see the contrast. What type of cartilage would hyaline cartilage, perfect. What you can see here even more is the basophilic capsule that surrounds each chondrocyte. It's made of more acidic glycosaminoglycans. That's why they bind more basic dyes and appear darker. The other matrix here is called Interterritorial matrix because it's among the territories of the chondrocytes, and it's homogeneous. That's why we call it hyaline. And where does it come from? Perhaps in which organ you might find something like that? Hyaline cartilage. Any idea you can add? Where can you find hyaline cartilage? Yes, the trachea would be a good example. The bronchi, the costal cartilage that attaches the ribs to the sternum, the articular cartilage in the knee joints. Okay, great. I like that. Another more advanced question about the red blood cells, hemoglobin, and white blood cells. Look at these statements, and what do you believe it might be? It might be true. So this is already mimicking some real-life problems where the histology of blood components is involved somehow. Okay, this takes more time because these are more complex problems for first year. So I see most students have already answered. Let us check it to move forward. So I'm locking this now. Newborns and infants have normally a lower white blood cell count than adults. It's the opposite. The reference values of leukocytes for adults is a range between 4,000 leukocyte per cubic millimeter and 10,000 leukocyte per cubic millimeter. But in infants, in children, it's pretty normal to have 13,000, 14,000, 15,000 leukocytes per cubic millimeter. So if you'll be working with children, with infant patients, you will adapt to the different range of the blood count. In pediatric medicine, they have different reference values than in adults, and it makes sense. The activity of immune system, it's considerably greater in the childhood than in adults. Your patient is a heavy smoker. You are very surprised by finding polyglobulia in his blood sample. Now, what is polyglobulia? It's finding more red blood cells per cubic millimeter than it should be. So instead of living with, for example, 5.5 millions red blood cells per cubic millimeter, the patient has 6.5 millions or 7 millions per cubic millimeter. This is polyglobulia, increased number of red blood cells. How is it linked with smoking at all? The formation of new red blood cells is stimulated by a hormone called erythropoietin, which is produced by the kidney, by the way, mainly. And the strongest signal to produce more erythropoietin and to promote formation of new red blood cells is tissue hypoxia. And smokers, heavy smokers, They live in permanent, the tissues live in permanent hypoxia because the carbon monoxide in the cigarette smoke is binding irreversibly to the hemoglobin, and therefore the hemoglobin cannot carry oxygen. So the peripheral tissues get less oxygen than expected, which is a stimulus for producing more red blood cells as a compensation of that. So in heavy smoking patients, you will very often find more red blood cells than in a non-smoking population. Your patient's stomach was removed due to cancer. He has suffered from repeated inflammation of both kidneys. He might develop anemia. Yes. Why the stomach? Some cells of the gastric glands that are in the fundus and in the body of stomach, namely the parietal cells of the gastric glands, they are producing a glycoprotein called intrinsic factor, which is necessary for being able to resorb the B12 vitamin in the ileum, in the small intestine. So if the stomach is gone or its mucosa was destroyed by inflammation, you cannot actually use the vitamin B12 even though you have sufficient intake of it. Your body is not resorbing it. And B12 vitamin, we know, is already linked with maturation of red blood cells. So yes, you need to think in advance and provide that vitamin to patients without stomach. And the kidneys, we already know that's where the erythropoietin is produced. So if the kidneys tissue is destroyed, the patient will be low on erythropoietin and also endangered by anemia. And that's precisely what a doctor should know and should anticipate. Okay, your patient complains. So this was correct. I'm glad you knew it. Some students knew it. Your patient complains of feeling tired. This might not be explained by finding 105 grams of hemoglobin per liter of blood. That's a typical situation. The patient says, "Oh, I'm so tired last days," especially when I'm doing sports, for example. And you start looking for an explanation. And one of the data you will get will be this. So you should know that the normal concentration of hemoglobin per liter of blood would be 120 grams per liter in female and 130 grams per liter in male. So yes, it might be one of the traces you should follow in your diagnosis, in your diagnostic algorithms. Especially this would work in patients who are physically active. They will notice even this slight decrease in hemoglobin. If the patient would spend the day like sitting in a couch, in a sofa and watching TV, it would not make him tired. Yeah, but if anybody's doing sports, they will definitely notice. And this is about the blood group inheritance pattern. If the mother is ARH negative, and the baby will have AB RH positive, and the husband is 0 negative. So yes, if the husband knows something about the group inheritance pattern, he might doubt if he's the father, because there is the phenomenon called co-dominance in the inheritance of blood groups in the ABO system. And for example, where did the baby inherit the B antigen from? Not from the mother, she didn't have it, and not from this father. Also, where did the baby get the RH antigen, the rhesus antigen from? Not from this mother and not from this father. Okay, so that's okay. And last, will be this one, and it's very simple. Name the leukocyte you see. What is this? So some students believe it's a neutrophil. Okay, some students believe it's a lymphocyte. It's a thrombocyte. Some students believe it's an eosinophilic granulocyte. Okay, well, there is only one leukocyte here, and it's this one, yeah? Okay. Okay, I got it. So this is definitely a neutrophilic granulocyte, or briefly, a neutrophil. How do we know? You need to focus on the morphology of the nucleus and the presence, absence, or the color of and size of the granules. So first, the nucleus. A lymphocyte, as suggested here, would never have this segmented nucleus. This nucleus seems to be segmented in two segments. One segment that is a chromatin bridge, and second segment. This is typical for granulocytes that have segmented nuclei. So once you see this nuclear pattern, you forget about lymphocytes, monocytes, you start thinking about granulocytes, which narrows the diagnosis, because we have three types of granulocytes: neutrophilic, eosinophilic, basophilic. Now look at the granules. You don't have a microscope here. If you had, you would use the fine focus adjustment to focus through the cell. You don't have the opportunity here, so you need to trust me. But there are only very, very small, fine granules here, and they are neither eosinophilic nor basophilic. Neither of those is in Latin called neuter, and that's why these are called neutrophilic granulocytes. They don't stain as eosinophils, neither as eosinophils nor as basophils, none of both. So this is definitely a neutrophilic granulocyte. This is the most common white blood cell in the peripheral blood. So now I got an idea. Now I know what most students do understand and what they don't. So the rest of the lecture will be about a revision of the most problematic parts we have encountered in our revision. And so today's lecture is not a typical one. We need to bridge the gap for the past weeks. And the next lecture in next week will be already the normal one. So I prepare these schemes that I usually draw in my lectures. And you should know about the classification of connective tissue, the cells, and the extrasolar matrix. So which cells for the connective tissue you should know? You are supposed to know fibroblasts producing extrasolar matrix. They have this euchromatin in the nucleus, or more differentiated form and resting forms fibrocytes. Also, there is a modification called myofibroblast, which is a fibroblast with contractile myofilaments. Okay, you should know adipocytes. There was no problem with that. The unilocular and multilocular adipocytes. These are the most common in the human body. The multilocular occur only mostly in newborns, in specific regions such as mediastinum or around the kidney. You should know the reticulocytes in the reticular connective tissue with the reticular fibers, and you should know the pigment cells that are releasing the the pigment granules. For the wandering cells of connective tissues, you should know the monocytes. By the way, I hope the sharing is still running. Just give me a short yes/no feedback if you see the schemes I'm giving comments on. Yeah, great, thank you. You should know the mastocytes. They have these granules with histamine and heparin. Everybody knows the function of histamine. It increases the permeability of our blood vessels. That's why after an insect bite, the tissue gets red because there's more, and there is edema because the vessels are dilated, and more permeable, so more fluid will enter the tissues from the blood plasma. And there is also the heparin, which is a natural anticoagulant. So upon various stimuli, these mastocytes might undergo degranulation and contribute to some inflammation processes. Macrophages, oh, there's a typo here, macrophages, they have these lysosomes which fuse with phagosomes, and after destruction, partial destruction of these substrates, the antigens are exposed on the surface, which serves as an activation signal for lymphocytes. This is called presentation of antigen, okay? And these cells are one of the antigen-presenting cells. We should also know that these macrophages differentiate from blood monocytes. They leave circulation, enter the tissues, and that's where they become macrophages. We should know the plasma cells that are producers of immunoglobulins. That's why they have this massive granular endoplasmic reticulum, which makes them basophilic. The only region that is not basophilic in the cytoplasm is the Golgi complex. So these they have this typical pattern of distribution of chromatin, which is used for identification of these cells in the slides. It's called cartwheel chromatin or clock phase. You should understand how the collagen is produced and organized. The most common amino acid here being the glycine, the smallest amino acid, which makes it possible for these molecules to come very close to each other and makes this triple helix of tropocollagen very stable. Tropocollagen units are organized with periodic overlap into collagen microfibrils. The fibrils are too thin to be seen with your routine microscopes, but they are organized into collagen fibers, which already you can see. And frequently, the type 1 collagen is even more organized. The fibers occur in bundles, and that's what you can see in your microscope. So why do we need vitamin C for collagen metabolism? Is because it serves for regenerating the enzymes producing this amino acids. Like prolyl hydroxylase and lysyl hydroxylase are the enzymes that are hydroxylating the proline and lysine amino acids in the sequence of the molecule. And without vitamin C, you cannot produce this hydroxylated amino acids, the hydrogen bonds of which are stabilizing the molecule. So without vitamin C, we don't produce a stable collagen, and that's why we can suffer from scurvy. Reticular fibers are, oh, I go back to collagen. There are more than 25 forms of collagen in textbooks. The students should be familiar with the first 5, yeah, where they occur in the human body and know the examples. By the way, everything is in the presentations that are on the Moodle. I'm just highlighting the key points in a graphic form, right? Yeah. You should know the reticular fibers, which are actually type 3 collagen. They are stained with silver that precipitates on the surface. And you should know the elastic fibers are made of an amorphous core surrounded by microfibrils of elastin. They are made mostly of hydrophobic amino acids that form these hydrophobic domains that prefer this relaxed condition. If you make it stretch, it's stretched, but once it gets opportunity, it will spontaneously relax. And that's the principle of the elasticity. Regarding the ground substance, you should know about the glycosaminoglycans, proteoglycans, and glycoproteins. Here is the basic composition explained. And the hydrophilic properties, if you remember. What type of connective tissue proper should you be familiar with? And you should be able to recognize once you see it in the tissues. It's the mesenchyma with mesenchymal cells, many capillaries, lots of ground substance. It's a mucous connective tissue with especially great fraction of ground substance, such as the hyaluronic acid, and every student should be able to provide an example, okay, where this occurs in the human body. The next, will be loose collagenous tissue and dense. To find out whether you are looking on loose or dense, think about the fraction of fibers or cells and ground substance. If there are more cells and the ground substance, that's the yellow component here, than fibers, it's loose. You can have literally any cell of connective tissue there because they might even migrate from the blood vessels into the connective tissue. It makes the lamina propria of mucosa, it makes the submucosa, for example. While in dense collagenous connective tissue, the fibers prevail. Either the fibers are organized in preferential directions, such as in tendons, ligaments, aponeurosis, and so on. You call it regular dense connective tissue. Or the fibers run in various directions, such as in the reticular layer of your dermis, or fibrous capsules of organs such as liver, kidney, thymus, lymph nodes, spleen, etc. And that's where you call it irregular dense collagenous connective tissue. Okay? You should know the white versus brown adipose tissue. The difference is the type of the adipocytes. And also the brown adipose tissue has more capillaries and it has this sympathetic nerve innervation, and being stimulated by sympathetic nerves, it produces heat because the chemical energy stored in the lipids here can be directly transformed to heat. There is a shortcut on the mitochondrial membrane. So instead of using this chemical energy released from the lipids to produce ATP, like it's usual here in white adipose tissue, it is directly used to produce heat, body heat. This is essential in newborns. Yeah. Elastic connective tissue is the one where elastic fibers prevail. They are highly interconnected, highly complex. An example here is the middle layer of largest body arteries like aorta or pulmonary trunk. So that's elastic connective tissue. Unfortunately, as we age, we are less and less able to produce sufficient amounts of elastin. That's why that's one of the reasons why our tissues get less and less elastic as we age. We are not able to replace the elastin in the same way we are losing it. And the reticular connective tissue, mostly you can find it in lymphoid organs. In this case, the reticular connective tissue is heavily populated by lymphocytes. And here are the examples: lymph nodes, wide pulp of the spleen, lamella propria of tonsils. Or the reticular connective tissue occurs also without the lymphocytes as the fine interstitial connective tissue of the lungs, kidney, and liver. Okay, the cartilage. Everybody should be able to understand the difference between these three types of cartilage: hyaline, elastic, and fibrous. So it's made of chondrocytes, they are grouped into isogenous groups, which are the daughter cells of the same mother cell that remain together. And this occurs in hyaline and elastic cartilage, these isogenous groups. There is a perichondrium on the surface of cartilage with the inner chondrogenic layer where you have new chondroblasts and the outer fibrous layer with the blood vessels. The cartilage itself does not contain blood vessels. I mean the mature cartilage. Yeah? The blood vessels are present only in the growing or ossifying cartilage in the embryogenesis or in the early childhood. But the mature cartilage has no blood vessels. It's avascular, which decreases the metabolism, but also it decreases the any regeneration. A tissue without blood vessels cannot regenerate pretty much. So once you got injury of the cartilage as an adult person, it's more difficult to heal. Elastic cartilage. The examples. Hyaline cartilage makes most of the fetal skeleton, persists in growth plates, persists as articular cartilage, trachea, bronchi, etc. The elastic cartilage, you have it in your pinna, in the external ear, try to bend it, it's flexible, right? Because the hyaline cartilage is fragile, but the elastic is really elastic due to these elastic fibers that make a network inside. And another example would be the epiglottis, also can bend, and part of the auditory tube, which is in the wall of the nasopharynx, where it elevates the torus tubarius. In these two types, the type 2 collagen prevails in the matrix, okay? We also have plenty of glycosaminoglycans and proteoglycans that bind lots of water, and it makes the cartilage pressure resistant, incompressible. The third type is kind of a mixture of dense, irregular collagenous connective tissue and cartilage. That's why we call it fibrous cartilage. The thick type 1 collagen fibers run in every possible direction, and the chondrocytes are rare. There are only a few chondrocytes there. You can find it in this tissue forms the external layer, the annulus fibrosus of intervertebral disc. It's very pressure resistant in tension and also in traction. The pubic symphysis, it's a typo here, it should be Y here, and the articular discs, such as the articular discs in your temporomandibular joint is made of this one, or the menisci in the knee joint are made of this. Let's revise shortly the bone tissue you have missed, I believe, 2 weeks ago. So what should everybody know about the cells and extrasolar matrix? The cells are the osteogenic cells that come from mesenchyma that differentiate into the other cell types. Then the osteoblasts, they are producing bone matrix. First, they are producing unmineralized osteoid that is later on mineralized. And the osteoblasts differentiate into osteocytes, which are already embedded within the matrix. And they have long processes that go through the whole bone tissue, surrounded by the interstitial fluid. So the osteocytes can actually perceive the pressure, the mechanical loading of the bone, and respond with their metabolism accordingly, because the pressure transmits the signals to the cells, so they can adaptively remodel according to the loading. And there are osteoclasts that are special macrophages that are able to resorb the bone. Now, this resorption by the osteoclasts and the bone formation of the osteoblasts needs to be balanced somehow. Yeah? Together, we call it bone remodeling. The matrix has an organic component and inorganic component. In the organic part, we have mostly type 1 collagen and some glycosaminoglycans. The inorganic part, the mineral part, is mostly made by hydroxyapatite, where some of the ions could be substituted. It's hydrated. A calcite is a minor component. Most crystals are made of hydroxyapatite and they are attached to the type 1 collagen of the organic part. From histological point of view, we have two types. The first that originates during embryogenesis is this one. The osteocytes and the connective tissue fibers, they run in various directions. That's why we call it fibrillar or woven or primary bone. This is remodeling to this histological type of bone that makes like 98% of our skeleton. It's called, lamellar or haversian or secondary bone. The osteocytes are organized in more or less concentric layers, and also the matrix is organized into concentric lamellae. This gives the bone tissue the ability to adaptively remodel in directions of the mechanical loading. So that's the key building principle of our skeleton, to make it pressure resistant and to constantly remodel according to the mechanical loading. This bone is not able to do that because it has the same properties in all directions. The fibrillar bone persists only in tuberosities, such as the tuberosity of the tibia, where the tendon of your quadriceps femoris muscle is attached to, or inserts, or the patellar tuberosity, where the Achilles tendon inserts. The rest of our skeleton is made of this. How the bone tissue could be organized? It could be organized into trabecular or compact bone. Trabecular bone is actually most of the bone of a human body, also called spongy or cancellous. We got these trabecules. On the surface, we can find the osteoblasts or osteocytes. Inside, there will be the haversian systems. And the cavities are filled with bone marrow. Either it's a red bone marrow or yellow bone marrow. Red bone marrow is a site of hematopoiesis in the postnatal life. In the compact bone, which is usually forming the cortical layer of our bones, sometimes it could be thick, as in the diaphysis of long bones, but the compact bone does not have these cavities, and it's made of densely organized osteons that are constantly being remodeled. So you can find traces of old or previous generations of osteons here. The surface is covered by periosteum. Again, it is a fibrous layer on the surface, which is attached to the collagen matrix of the bone, and it is the inner layer with the osteoprogenitor cells that are important for the healing of bone fractures. The bone tissue originates in two ways in the human body: desmogenous or chondrogenous ossification. The desmogenous is a direct differentiation from mesenchyma. So some mesenchyma cells become osteoblasts and they start to produce the bone matrix. Which bones originate like that? It's the flat bones of the skull vault, it's the upper and lower jaw, and part of the clavicle. All the other bones in your body are based on cartilage models that later on ossify. You should know, or students should know, the zones of this process called endochondral ossification, how the hyaline cartilage proliferates. That's the growth zone of that growth plate. And it's calcified for a short time, but then it's destroyed to make space for the newly produced bone trabecules, which are produced by osteoblasts. So these are the zones of the endochondral ossification. And well, which bones originate like that? Simply said, all the other bones of your human body except these. Yeah? Is the prevailing mechanism of bone formation in our body. And here is the chondrogenous ossification. Every bone in your body has some ossification pattern. It strongly depends on the invasion of blood vessels that help to destroy the cartilage, bring in new cells, and form the bone marrow cavities. So you can see it on X-rays, these ossification centers, as they appear during the age. What remains from these cartilaginous models is the growth plate cartilage. In women, in lung bones, it's like until the age of 16 to 17, where the bone growth is arrested. In males, it's 1 or 2 years. It takes like 2 years longer. So around the age of 20, the bone growth is normally arrested. Is the side effect of the puberty, of the androgens, the sex hormones that are, on one hand, they are accelerating the bone growth, but are also initiating the closure of these growth plates. And another cartilage that remains from this cartilage model is the articular cartilage. Yeah, articular cartilage does not have perichondrium, of course. It wouldn't survive here due to the friction. The nutrition of the articular cartilage is not provided by perichondrium, but from the inner layer of the fibers of the articular capsule, from the so-called synovial capsule and synovial fluid. Now we go to the blood, which was the topic for this week, and the blood formation. So here I try to summarize the key points of the blood. When you will read it in your textbooks and you will feel lost, you can go back to this slide. It's useful to know that it makes like 7 to 8% of your body weight. You know these units from biophysics, perhaps, because every lab you will send your blood samples to, well, samples of your patients, will provide the results in different units. Some provided in per microliter, which is the same as cubic millimeter. Some labs provide the results calculated per 1 liter, so you should be able to calculate that. We go to the liquid part. The students should know that the plasma is different from the serum, because serum is what remains from the plasma, is the liquid part after coagulation. So serum is plasma minus the components consumed during coagulation, which is mostly fibrinogen and other blood clotting factors. You should know that because, again, in biochemistry, lots of results are expressed or recalculated per serum or per plasma, and it might be different figures. Okay, like glucose per plasma or glucose per serum is different. So this is labeled by this P or S in the lab results. Then we have form blood elements, the proportion of which in the full blood is called hematocrit, is the fraction form blood elements divided by the full volume of the blood. The red blood cells prevail here. You have the reference values for male and female. Then we got blood platelets. Here are the reference values. Leukocytes, also known as the white blood cells. Here's the normal range in adults. And now we care about which leukocytes are present. So the most common are neutrophils. Less common are eosinophils and basophils. The second most common white blood cell is are lymphocytes. They all look the same in the routine sections. And if you need to classify these in immunology, you need to use some surface molecules to detect specifically these types of lymphocytes. These are T lymphocytes divided to T cytotoxic, T helpers, and T regulatory. There are B lymphocytes that differentiate later on, once activated, into plasma cells that are producers of immunoglobulins, and they are natural killer cells. Last but least is the monocytes, the largest cells of the peripheral blood that differentiate into tissue macrophages once they leave the blood. This is how to produce a blood smear. A droplet of blood is spread on the slide and observed during using the all immersion objective to increase the resolution. You find a systematic way how to meander to the slide to count as many cells as possible. And here are the foreign blood elements you should be able to identify. Every student should know how these foreign blood elements look like. My recommendation is look at the nucleus, at the shape and segmentation of nucleus, and look at the granules. And by combining these two criteria, you can easily tell one white blood cell from another. So you should be also familiar with the fact that the neutrophils, as they age, they increase the number of nuclear segments, which is used in the medicine because the younger forms might prevail in patients undergoing acute bacterial infection. The older forms might prevail, for example, when there is some problem with producing new neutrophils. Eosinophils have this typical bilobed nucleus and large reddish granules. Basophils, they are hiding their nucleus, which is irregularly segmented. Is hiding behind the huge conspicuous basophilic granules. So these are granulocytes. Because of the complicated morphology of the nucleus, they are also called polymorphonuclears. Unlike the agranulocytes, the lymphocytes, I got the question, can you find macrophages, not monocytes, in the bloodstream? No, macrophages are typically tissue cells. You can find these in tissues where they perform phagocytosis. Okay. Lymphocytes. In peripheral blood, you usually find small, medium-sized, or large lymphocytes. The small being the naive lymphocytes that haven't encountered or interacted with antigen yet. The large are usually the activated lymphocytes or natural killer cells. They tend to be larger. But look at the nucleus. It's large, almost spherical, and there is few cytoplasm. The only granules you can find here are so-called eosinophilic granules, which are basically lysosomes. They are present also in the granulocytes. And the largest cell in the blood is the monocyte with a kidney-shaped nucleus, lots of cytoplasm. Here is a detailed classification of the granulocytes, because especially the lymphocytes require more explanation. This is only for the purpose of histology. Once you will discuss this in physiology classes, in the physiology of immunity in the second year, or in the immunology, you will see it's much more complicated. But for histological purposes, you need to differentiate B cells that become plasma cells or memory cells. They contribute with antibodies, which you called humoral immunity, and T cells that differentiate in the thymus, namely the T helpers. They carry this surface molecule CD4. The T cytotoxic that are killing target cells, they carry the CD8 surface molecules, and T regulatory that try to orchestrate the response together with the T helpers. And they are actually more types. The natural killer cells are able to kill cells similarly like the T cytotoxic, but they don't need previous activation by some antigens. They are just scanning all the cells they come in touch with. And if they don't find the proper molecular identity, which is the human leukocyte antigen group of molecules or major histocompatibility complex, class 1 molecules, they kill these cells. Which cells have a modified molecular identity? Mostly cells that have been infected by some virus, or cells that undergo tumor transformation and suffer from some mutations they were not able to repair. So they need to be eliminated. We should remember that natural killers are important cells in antivirus immunity, and anti-cancer immunity. Now, the hematopoiesis. Per every kilogram of the body weight, every day, huge amounts of foreign blood elements are produced in our body. So it's an important process. It's a matter of life. It's really important. It all starts with the multipotent stem cell, hematopoietic stem cell, often abbreviated as the HSC. It's a stem cell, so it undergoes self-renewal during the whole lifetime. But some of the progeny differentiates. As far as we understand it by now, there are two main lines: lymphoid line and the myeloid line. So the lymphoid stem cell differentiates during the process called lymphopoiesis, formation of lymphocytes, and at the end of the process, we got lymphocytes. The myeloid stem cell differentiates into three more types of progenitor cells that start three different processes. The progenitor cells are abbreviated as colony-forming units. That comes from the lab speech, as the people who are handling the cells and cultivating these cells are used to communicate. They named it CFUs, colony-forming units. It comes from the way how they are handled and examined in the lab. So this progenitor cell starts the erythropoiesis, which after some stages produces red blood cells. This progenitor starts the formation of thrombocytes, and this progenitor has two options: either enter granulopoiesis to form granulocytes, neutrophilic, eosinophilic, basophilic, or monopoiesis, producing monocytes. And now I will go briefly through each of these lineages, right? Starting with the erythropoiesis. So we already know this beginning, and let's go further. The progenitor starts to produce RNA. That's why it needs nucleoli for. We call it pro-erythroblast. Next stage has really plenty of granular endoplasmic reticulum, which makes it basophilic. We call it basophilic erythroblast. On this granular endoplasmic reticulum, the hemoglobin will be produced in the next stage. The hemoglobin protein or hemoprotein is already eosinophilic. So this next stage is partially basophilic due to the granular endoplasmic reticulum and partially eosinophilic due to the hemoglobin. We call it polychromatophilic. Poly means more or many. Chroma is color, and philic means it likes it. So it likes more colors. It's a polychromatophilic erythroblast. And in next stage, enough hemoglobin has been produced, the granular endoplasmic reticulum is lost. That's why the color is already eosinophilic. And it's called eosinophilic, or a synonym is orthochromatophilic. Orthos means like the right, the correct. Chroma is color, and philic, it likes it. So it already likes the right color of the future erythrocyte. So that's the strange word students refuse to remember, but it's part of the official terminology. So it's the last stage where the cell has a nucleus. The next step is the extrusion of that nucleus, which is swallowed by the macrophages, by the surrounding tissue macrophages. And we got a reticulocyte without the nucleus, but with some remnants of these polyribosomes in the cytoplasm. This cell already might enter the circulation. But in the peripheral blood, the mature erythrocyte prevails. If you look on this staining pattern, the terminology is based on, it could be easily explained by the decreasing amounts of ribosomal RNA, RNA, and increasing amounts of hemoglobin during that process. So that's the erythropoiesis. It's stimulated by the erythropoietin. I believe we mentioned it already. It's made in the kidney, and the strongest stimulus for producing more erythropoietin and promoting this process is tissue hypoxia. Okay. The next lineage, we go back, will be this thrombopoiesis. Okay, in detail. The thrombopoiesis from this progenitor proceeds to a megakaryoblast. These cells undergo a process called endomitosis, where the nuclear content is multiplying, but the cell is not. So we got multiple nuclei. It's a polyploid cell called promegakaryocyte. It matures into a megakaryocyte, which sits in the bone marrow. It has many nuclei, and it's sending these long branching processes into blood vessels of the bone marrow. These bone marrow capillaries are also called sinusoids, and they have gaps between the endothelial cells, which let this process easily in. And once in the contact with the bloodstream, these processes become fragile, and they are fragmenting into fragments we know as thrombocytes. So the blood platelets are just fragments of the cytoplasm of the huge mother cells called megakaryocytes. The branching processes are also therefore called proplatelets. Now I go to granulopoiesis and monopoiesis. So I go back to my overview scheme, and we follow now this lineage, okay? Granulopoiesis and monopoiesis. So first, granulopoiesis. The stage here is myeloblast, differentiated into promyelocyte, which already has some acidophilic granules. The next stage is called myelocyte. It starts to express the specific granules, namely neutrophilic, eosinophilic, and basophilic granules. That's why we have 3 types of myelocytes. The specific granules first appear only in a limited region of the cell. The next stage comes after the myelocyte, which is expressed by this prefix meta. Meta means like behind something, next or after something. So the cell that comes after the myelocyte is metamyelocyte. And we got 3 types of metamyelocytes. They have already the granules distributed more evenly. We have neutrophilic, eosinophilic, basophilic, metamyelocyte. And the final form that already enters the bloodstream are the granulocyte, as you know these from the blood smear. The youngest neutrophils have an unsegmented nucleus. That's why they are also called band cells or step cells. But as they are growing older, the nucleus becomes segmented to 1 to 2, 3, 4, and up to 5 nuclear segments. But you already know these cells from the peripheral blood. I go back from the same progenitor. The monocytes are differentiating. It's another part of that lineage. It starts with monoblasts, pro-monocyte, and a mature monocyte that is in the peripheral blood. After entering tissues, it becomes macrophage. The granules you can find here are the lysosomes, actually. And last but not least, I will follow this lineage, the lymphopoiesis, that will lead us to lymphocytes. And with some simplification, we have a lymphoid common progenitor, which can differentiate into B lymphoblasts or T lymphoblasts or precursors of natural killers. And the B lymphoblasts are differentiated into B lymphocyte, T lymphoblasts into T lymphocyte, and natural killer are on their own. The B lymphocyte, after entering the tissues, after being activated, they differentiate into plasma cells, the producers of immunoglobulins. Now you might ask, where does it happen in the body? It depends on the age. If you follow this axis, it shows the age. Here will be the birth. Okay, the prenatal hematopoiesis is here labeled in months. in months of the prenatal development, and the postnatal life is in tens of years. So I start with the first hematopoiesis. It starts in the mesenchyma, which is the embryonic undifferentiated connective tissue, or in the vicinity of structure called the yolk sac. It's one of the structures of a early embryo. That's where the first mesenchymal cells turn to angioblasts, which are the precursors of circulating cells, and the first cells start to circulate and carry the oxygen. In the third month, this hematopoiesis, the blood formation, migrates into the liver. In the meantime, the liver becomes the largest organ of the fetal body, and among the trabecules of hepatocytes, liver cells, and in the wall of the sinusoids, of the liver sinusoids, liver capillaries, there is this hematopoietic tissue. Also, the spleen helps a bit, but spleen is much smaller than liver, so this tries to approximate the capacity of the hematopoiesis at the age. So we call it together, this period is called the hepatolional hematopoiesis. We need that because we don't have any bones, we don't have any bone marrow at that age. Once the bones are formed, the bone marrow cavities are growing, the hematopoietic mesenchyma migrates into red bone marrow. So there is a gradual increase of the hematopoietic capacity of the red bone marrow. And at birth, it's already the bone marrow, or before birth, it's already the bone marrow that predominates. Until the age of 10 or 20, the bone marrow is distributed in most bones of the human body. But the capacity is related to the anatomical size of these bones. So I put these long bones that have a great capacity for hematopoiesis, but soon the red bone marrow will be replaced in these long bones by yellow bone marrow, which is just adipose tissue. And here are the bones where the hematopoietic bone marrow persists in adults. So it's in the bodies of vertebrae, in the pelvic bones. Pelvic bones are huge bones with lots of red bone marrow. Flat bones of the skull vault, ribs, and sternum. So a doctor should know where to get a sample of red bone marrow in adults. So either from the sternum, it's done, or from the pelvic bones. Now the bone marrow is amazingly complicated ecosystem with so many growth factors, molecular differentiation factors, and so on. So this is only a oversimplified sketch of structures you can expect there in the real slides. So first I will mention the special blood vessels that are highly permeable blood vessels called blood sinusoids that have openings among the endothelial cells that let cell processes or whole cells easily enter the bloodstream. We call these capillaries blood sinusoids. So you can see here one of the megakaryocytes producing the platelets here. You can see the such a nest of erythropoietic cells with different stages of the erythropoiesis that are releasing reticulocytes or red blood cells into the bloodstream. Okay. Next, there will be a similar islet for granulopoiesis and so on. They are not included all in the scheme. The stroma that is maintaining the structure of the bone marrow is made of reticular connective tissue. So we got reticular connective tissue cells and some fibroblasts and some adipocytes. The cellularity of the bone marrow decreases with age. So if you have noticed in the graph, this decline in the capacity of the hematopoiesis So every year you're older, your overall capacity for producing blood cells is slightly declining. That's it. So I would like to show some more pictures in a presentation. If you've got any questions, just put it in the chat. I will answer it at the end. But I want to show some real pictures. I hope you'll understand these now. The presentation is already online on Moodle. So this is the real red bone marrow, how it looks like. You can see the bone trabecule here, and here is the red bone marrow with the hematopoietic islands. This is one of the vascular sinusoids into which the newly formed blood elements are released. These large cells where you can see more nuclei are the megakaryocytes, the producers of platelets. These are adipose tissue cells, adipocytes. Okay. This is another picture of bone marrow, again with the megakaryocytes. These are the most conspicuous cells there. You have two ways how to examine the bone marrow. Either as a liquid bone marrow smear, which resembles venous blood in its consistency. You can get it by a needle, aspirating it mostly from the sternum, which is closely to the body surface, so you won't hurt your patient too much. And it's examined on the histological slide like a blood smear, and you can scan it if there are some irregular cells that don't belong there, and the specialists that are trained to do that are able to say, okay, this bone marrow is okay and this one is not. If you need to examine the whole architectonics of the bone marrow, including the spongy bone trabecules, which gives you more information, you need to cut a whole cylinder of bone marrow with a very thick hollow needle. This is usually done in the pelvic bones of the iliac crest, where the hematopoietic bone marrow is close to the body surface. So you will cut this cylinder of bone and bone marrow, and you will examine it under the microscope. It shows you the proportion between the lineages, if there is some, for example, increase in the fraction of connective tissue that is suppressing the hematopoietic tissue. That could be one of the diagnoses you can make out of it. This schemes shows you all the lineages at once. We already went through that one lineage after another, but this is a complex view on it. This shows you some of the important points, such as nuclear extrusion, by which the reticulocyte originates, and some other real cells, because my schemes were simplified. Okay, this is bone marrow. This is the region where the erythropoiesis happens. This is the erythropoietic island, and this is the granulopoiesis. Okay, so it really needs some expertise to tell what is what. Perhaps before you get to the 5th year and you'll be dealing with hemato-oncology, the explanation will be different, because we don't understand the hematopoiesis by now completely. Our understanding is not a complete one, so ask your teachers when we're dealing with hemato-oncology. Let me also mention the bone marrow transplantation. Typical recipients are patients suffering from leukemia, lymphomas. These are malign diseases of the hematopoietic tissues. So it's leukemia or tumors of lymphoid organs, lymphomas are solid, these are liquid tumors. Patients with some hematopoietic disorders, yeah? The donors should be of good health, and this might vary, the criteria may vary country from country. Make sure you'll find out how it's organized in your country or where you'll be working. In Czech Republic, these are the criteria. And a donor, a volunteer donor, undergoes typization of the human leukocyte antigens, that's the molecular identity on the surface of the cells. And there's a database that tries to match the donors with the recipients to find an optimum match between these two. The own procedure how to get the bone marrow once a suitable donor is found can be done in two ways. The classical one, where the bone marrow is harvested under general anesthesia. You can see they're using syringe to suck the bone marrow in from the pelvic bones. Or newly, you can use some factors that you inject into the patient's blood, and they will provoke the stem cells to migrate into venous blood. So it's like much less invasive. And each country has its network of registers. Pilsen is the key organizer in Czech Republic because that's where it originated, the whole bone marrow transplantation program in the early 90s. And it's spread through the country. And these days, most countries cooperate with each other when they are not able to find a suitable recipient or donor in their own population. They are exchanging the samples. A few words about the blood donation. Again, please make sure how it's organized in your country. If you have time, I'll be curious if you could type it in the chat. There's a question, by the way, where can we find the recording of this lesson? The presentation is online. I will show you in a minute. And I'm also trying to record this. And if I succeed, because it's my first time I'm recording this, it should be on the YouTube channel of our department. Okay. By the way, if you can find out how the blood donation program is organized in your country, the criteria are different. In most countries in Europe, as far as I know, it's on the voluntary basis because it's the less risky way how to do that. You don't want to attract people who are looking for money for blood donation because this might be people with increased risk of diseases that could be transmitted to the recipients. And the safety is here the rule number one. Yeah, safety, and again safety of both the donor and the recipient of blood. So who are the people that need the blood derivatives? People with major traumas, undergoing major surgery. They often, if this is a planned surgery, they undergo so-called auto-transplantation, where their own blood is stored and they get it back when they need it. And people suffering from hematologic disorders, hemophilia, leukemia, anemia. The usual volume of blood that is taken is 400 to 450 milliliters. In Czech Republic, the women are allowed to donate like 3 times per year. In men, it's like 4 times per year. It also depends on the body weight and other criteria. Nowadays, the full blood that is taken is processed to get as maximum of the components as possible. You don't want to waste anything from it. So if your patient needs red blood cells, you are not transfusing the whole blood because the patient does not need the platelets for example. Another patient might need the platelets, or if the patient needs some coagulation factors from the plasma, he or she doesn't need the red blood cells at the same time. So every component is processed separately. It has also different or various expiration date, because the red blood cells, the half time is like 100 days. The lifespan of red blood cells is like 100 to 120 days. So the half time is like 50 days. Okay. The lifespan of platelets, of thrombocytes, is like 10 days. So the half time is like 5 days. Okay. And the frozen plasma might be stored for a much longer time. So after processing this, you give your patient what he or she really needs, only that component and nothing else. Okay. Now you can test yourselves briefly. I got an overview, and that might be my final part of the presentation. Few real pictures from the peripheral blood smear. So how to make diagnosis here? I see this, and this. It's a cell with a size comparable with a red blood cell. So it's a relatively small cell. It is a round nucleus or nearly round nucleus, only a few cytoplasm. These are lymphocytes. Okay. While this cell, for example, with this indentation of the nucleus, with this incision, it is much more cytoplasm. It's large. It's like 1 or 2 red blood cells. It's the monocyte, and so is this one with the kidney-shaped nucleus. So these are monocytes and these are lymphocytes, together called agranulocytes. Here we got some granulocytes. This one and this one has different nuclei, but it's a neutrophil. I can tell because of the granules. The granules are small and they don't have any conspicuous color. So this is a very young neutrophilic granulocyte, and this is an intermediate, let us say, neutrophilic granulocyte. By the way, this appendage here might be the Barr's body, one of the inactivated X chromosomes, probably. I would need a more closer look. This one has 2 nuclear segments and huge reddish granules. That's typical for eosinophilic granulocyte. Again, huge eosinophilic granules. This is eosinophilic granulocyte. These two, I can barely see the nucleus. It is an irregular form, but is hiding behind the dark, huge basophilic granules. That's why this one and this one is the basophilic granulocyte, right? And more forms. The cells of the blood are highly variable, so we need lots of training to find out. This is a huge activated lymphocyte. I can tell according to the round-shaped nucleus and the eosinophilic granules here. This is already a monocyte, kidney-shaped nucleus, eosinophilic granules, and the cell size is greater. This is a neutrophil. It's a young neutrophil, is a so-called step cell or step cell. I can see there are fine neutrophilic granules in the cytoplasm. By the way, these particles here are thrombocytes. They vary in size, like from 2, 3 to 4 micrometers, usually 2 or 3. And you can see they have a darker region in the center. There's a granulomere with the granules that are released upon activation to promote the blood clotting. And they have a more peripheral lighter zone. It's called hyalomere. Yeah, these are platelets. This is a platelet. This one. This would be a granulocyte with basophilic granules, so it's a basophilic granulocyte. This shows you a contrast between a lymphocyte with round nucleus and a neutrophilic granulocyte with 1, 2 segments. This is a neutrophil with 1, 2, 3, 4 nuclear segments, so it's an older one. This is a monocyte. Again, this is a lymphocyte with a round-shaped nucleus compared to a typical monocyte with its incision inside the nucleus, a large more cytoplasm, and it's a large cell compared to cyte. Monocyte, lymphocyte. Again, this is another neutrophil. This is another neutrophil. This is another neutrophil, a young one. This is a lymphocyte. I believe we know why. And here we go, lymphocyte, monocyte, neutrophilic granulocyte. 2 lymphocytes, eosinophilic granulocyte. I see the reddish granules. And that's it. So now I go to your questions, if you got any. Can you explain the credit system again? What do we need to get the credit? This might differ from group to group, but I still need you to be a little bit patient because we are waiting for the approval of the modified study rules that need to be approved by the university, because you are legally still studying in the present study form, and the credit exams were supposed to take in person. Now we understand that it's not sure if you will be able to take the exam in person, so there needs to be some change in the legislation. As soon as this is available, is done in these weeks, we'll inform the students. But I believe the best way how to prepare will be use our online test. I will show you where on the Moodle. So I go to the Moodle, Theoretical Institutes, Histology and Embryology, and yeah, preparation and training tests. So once you get in, there are the topics prepared for the first-year students, which includes cytology, epithelia, connective tissue, proper cartilage, bone, blood, muscle, and nervous tissue and nervous system. That's for the first-year students. So there will be some criterion for you passing these topics. Yeah, you can train now. You can take it by now, and we got a list of students, of users. I can check who by name did meet the criteria. I think like about 80% at least, because you have time, you have your textbooks, you can do it from home. So I believe at least 80% would be a good criterion. And second, if there would not be an opportunity for you to take the credit test in person due to a quarantine or whatever the situation will look like in your country and in Czech Republic at the end of semester, I can think about showing you a slide via a shared screen, for example, something like that. Yeah, I can show you a slide from a virtual microscope, and we can zoom in and out, and I will tell you, I will ask you, okay, what type of tissue is that? You will tell me, oh, this seems to be a connective tissue. Okay, what type of connective tissue? It's a loose connective tissue. How do you know? Because I see the ground substance, which is white here, and the cells, they prevail over the fibers. Correct. What else do you see here? I can see blood vessels here. These capillaries oh, this is a capillary. What else? You can see nerves here. So this is a mucous connective tissue, extremely loose connective tissue. And we move on, we focus as if we were working with a microscope. You'll describe this cells as odontoblasts. This is the dentin. These are dentin, this is the dentin with the dentin canaliculi, each containing a process of odontoblast. I will ask, what is this region of the dentin? You can say it's called predentin. It's an unmineralized form of dentin. I say, okay, and so on. We can make a short discussion as a slide identification. This might be one of the technical options we are prepared for, but please stay on hold. We're waiting for the rules to make it legal. Yeah. Where can we find a recording of this lesson? I hope it will be on the YouTube channel. So if you go to our histology department web pages, you switch to the English version. There is a link here to the YouTube channel. Yeah, you click on it, and you look on the playlists. And there are playlists in Czech, but they are also playlists in English. So on this histological schemes playlist, there are all the schemes for the whole semester, starting with the beginning of the semester with today's schemes, because that's our usual way of working with students. They actively participate, and we explain it step by step. You know, all the processes. We give our comments on it. So you got it all with some comments. So it's already until the end of this whole semester. So that's another resource you have. If I go back to the Moodle, the presentation will be here. The presentation of today's lecture will be here in the course Supporting Materials and Resources. And here are the presentations from the beginning of semester, and it includes today's presentation, right? So that's all the questions I got in the chat. And if there are no more questions, you can unmute yourself if you wish. We can discuss anything. Will the slides used for the credit test exclusively be those that are on the list? Yes. Yes. You are first-year students, so your credit test will be based on the first-year slides, because there are more slides waiting for you in the second year. Yeah. If you're studying from various textbooks, make sure you remain compliant with our educational goals. So if I go to the web page of our department, the English version, here in the education tab, you got the learning goals. And it's learning goal objectives for first year histology. It summarizes what you should be able to understand from the lectures and from the practical classes. All the words, all the terms. So if I, for example, take today's lecture, it's summarized on one page. Which terms should be familiar to you? Okay, what you should be able to do, explain, which answers you should be able to explain. So you can find the answers in most textbooks. We don't care actually which textbooks you prefer. Of course, there is a recommended list of literature, but most of the textbooks are sufficient. They are good. Regarding this topic, blood and hematopoiesis, I would like to point you to one more document that's online. It's into education among the study materials, and it's here the reference values of the blood smear. Yeah. Although the reference limits might slightly vary country from country, lab from lab, textbook from textbook, we don't care about some minor deviations. When you will be working as doctors, you will request these values from the lab you will be working with. Okay, but you should not hesitate if there are, I don't know, if there is 50 grams hemoglobin per liter normally, or 140. So make sure you know these reference values of healthy individuals, because you will need these definitely in so many parts of medicine. So that's another resource for this particular week. And starting with next week, we are already proceeding with the regular classes, and there will be muscle tissue next week. I'll look for more questions. Okay, there are no more questions. So my last point, that is already voluntary for you, so only students who would like to stay, if you got time, I have prepared some microscopic pictures just to replace or substitute for some of the practical classes that were lost. And I would like to give some comments on some microscopic pictures, revising the connective tissue, similarly as we revise the blood for today. So I say goodbye to the students who don't want to stay, and those who are interested in. This picture shows you a very loose connective tissue with fibroblasts, with some fine collagen fibers, and this is mesenchyma. It's embryonic connective tissue. It might be like slightly condensed here. That's typical for mesenchyma. This is the more loose form of it, but that's how mesenchyma looks like. It contains lots of blood vessels, such as this one, or this one. Next, this is connective tissue that contains lots of hyaluronic acid. These are these white spaces. It's a so-called Wharton's jelly from the umbilical cord. The cells here are mostly fibroblasts or fibrocytes. Those with a more compact nucleus, which is sharp on the one end, it's fibrocytes. Like here, those with a more spread nucleus are fibroblasts. So this is the Wharton's jelly from the umbilical cord. You have heard about reticular connective tissue. You have seen the scheme of it. This is how it looks in reality when you stain the reticular fibers black. This is from a lymph node, and you can see the reticular fibers make a network which is supporting the lymphocytes, which are mostly the round shaped cells among these fibers. This is to illustrate the dermis of the skin, which is made of connective tissue where these collagen fibers prevail. This eosinophilic substance is all type 1 collagen. There's also elastin, which are these black fibers. So it's dense collagenous connective tissue. And you can see the collagen fibers and the bundles of collagen fibers are arranged in all possible directions. So this is the irregular type of dense collagenous connective fibers. That's the reticular layer of the dermis. We have also the surface papillary layer, which is more less, I would say, loose collagenous connective tissue, and this will be the epidermis with the stratified squamous keratinized epithelium. This is definitely dense collagenous connective tissue because the prevailing component here are these thick bundles of collagenous fibers. Okay, you can see the nuclei of some perhaps fibrocytes, fibroblasts. This is a mastocyte, mast cell, heparinocyte. This is a loose collagenous connective tissue from a core of intestinal villus, and you can see how it's infiltrated by mast cells, by neutrophils. So next to fibroblasts and fibrocytes and capillaries, you can find, and plasma cell, this is probably a plasma cell, you can find all the possible wandering cell of connective tissues inside. This is the middle layer of the aorta. This is how elastin membranes are organized. So you can see a whole series of elastic membranes that are concentric, arranged in a concentric way. They are connected by thinner elastin bridges. So the whole system is made of repeating lamellae. One of these plates is called lamella. They have openings called fenestrations through which the vascular smooth muscle cells are penetrating. There is also collagen here, type 3 collagen. This is the white and brown adipose tissue. Interestingly, young adipocytes called adipoblasts also have this more vacuoli. This is cartilage. It's hyaline cartilage, and we can see that the cells are still dividing here. The chondroblasts are still dividing. We don't see the final isogenous groups formed yet because this is an embryonic cartilage, embryonic hyaline cartilage. The mature form of cartilage will have much more extrasolar matrix, which will be produced by the chondrocytes. And that's it. Both these examples are showing the hyaline cartilage in different stainings because the appearance strongly depends on the composition of the extracellular matrix, which types of glycosaminoglycans are there, and which staining are you using. So both is hyaline cartilage. We can already see the isogenous groups here. This is a nice hyaline cartilage from an embryonic preparation. It shows you that the embryonic cartilage does contain blood vessels. These are actually blood vessels that probably take part on the growth and of this epiphyseal plate, of this growth plate, where the cartilage is proliferating, the cells are increasing in size, and then is destroyed and replaced by the newly formed bone trabeculus in this growing bone. This is a joint, and that would be the epiphysis of another bone. I believe it's the knee joint of a fetus. So that would be one of the condyles of the femur. That would be the tibia, and this would be the patella. So the embryonic cartilage does contain blood vessels. The mature cartilage is avascular, without blood vessels. This is also a cartilage, we can tell by the chondrocytes and the small isogenous groups, but in this time it contains elastic fibers which form this brown or nearly black network here. It's from the epiglottis. Oh, sorry, it's from the external ear. It's the elastic cartilage of the external ear. The green substance, by the way, is mostly collagen. And this is how fibrous cartilage looks like. The prevailing component here is the collagen fibers. It's the type 1 collagen that forms the thick bundles. It has this wavy appearance, and you can see only individual cartilage cells, the chondrocytes, that are scattered among the prevailing collagen. So this is how your intervertebral disc looks like, namely the outer envelope of it called the annulus fibrosus. The chondrocytes are not forming isogenous groups. Only rarely you can find two chondrocytes together. Otherwise, the collagen prevails here. This is ossification. It's the ossification that originates directly from the mesenchyma. This is mesenchyma. This is also mesenchyma. And some of the mesenchyma cells become the osteoblasts. Here, these cells that are arranged on the surface of this bone trabecule, these are osteoblasts. Yeah? And they are producing the bone matrix, which first is not mineralized, but later on it becomes mineralized. And once the osteoblasts will form enough matrix, enough bone matrix, they will stay inside. They will be embedded in the matrix they have produced by themselves, and they will later on differentiate into osteocytes. You can see also this huge cell that is digging here. It's probably an osteoclast in a resorption lacume. So this is a newly produced bone trabecule. It's in from the embryonic ossification of the upper jaw, of the maxilla. Well, let's compare it to a completely different mechanism called endochondral ossification. We can see the growing hyaluronic cartilage, then the increased cells, then the matrix becomes calcified for a short moment, but then the cartilage is eroded, is destroyed via the cells called chondroclasts. So more space is made for these newly produced bony trabecules, and the cells on the surface that are producing this bone tissue are osteoblasts again. So these are the zones of the chondrogenous ossification. And this is already a mature bone tissue. It's a compact bone. This is a picture from the diaphysis of a tibia. So here would be the periosteum that has been removed. And here is the compact bone. You can see the vascular canals, the most common being the Haversian canals, each being in the center of a Haversian system, or osteon, which is the system of concentric lamellae. But you can see also vascular canals that connect one Haversian canal with another. This is called Volkmann's canal. The spelling is Volkmann, Volkmann's canals. So the Haversian canals are connected with other Haversian canals via the Volkmann's canals. The whole bone is interconnected with these vascular canals. The bone is not a static tissue. The bone tissue is a continuously remodeling tissue with high metabolic rate, and you can get an idea about how well it's perfused. How rich is the capillary bed here? And this is detail of some of the osteons. You can see the bone cells, the osteocytes, are sitting in cavities called lacunes, and they have these processes. They look like tiny spiders. They are sending their processes into the bone canaliculi. So the overall surface, the total surface of all these canaliculi is immense. So the bone cells are continuously taking part on the mineralization or remineralization or demineralization of the bone matrix. Here are lamellae that do not belong to any of the current osteons. It's because the new osteons are replacing the previous generations of the osteons. These lamellae that do not belong to any present osteons are called interstitial lamellae. And this is a high magnification of the osteocytes with a projection. So you can get an idea how densely the bone tissue is penetrated with the bone canaliculi, the microscopic bone canaliculi. So that was the short revision of the connective tissue. And if there are no more questions, then thank you very much for cooperating, and see you or hear you next week in the same time we'll discuss the histology of muscle, and it will already be an like regular lecture where we will focus on muscle itself. If you got any questions in the meantime, prepare your questions, and I'll be happy to answer this in the chat. So goodbye for now.